All right, well, it's a real pleasure to introduce today's speaker, Paul Schmidt from the University of Pennsylvania. For those coming in, there's a couple of seats up here. Paul received his PhD at Brown University, working with David Rand, and then he did a postdoc with Walt Eanes at Stony Brook before joining the faculty at the University of Pennsylvania, where he's risen through the ranks and has been ever since. So Paul is a remarkable scientist who combines the power of Drosophila as a model system with an interest in ecology. And so for those of you who think of Drosophila as being this one strain Oregon R, or who think of Drosophila as being this laboratory model organism that only exists in the lab, well, it turns out it has real ecology, and Paul studies that ecology, and it's remarkably interesting. And it's been, I should say, it's been a huge influence on my own work. So as many people probably know, Drosophila melanogaster is native to originally Africa and then Europe, and much more recently the Americas and the rest of the world. And there's clinal patterns of variation along the east coast of North America that reflects adaptations to different climates. And what Paul has done is work out a lot of the details of that. And he has a strong interest in life history, evolution. And he published a paper, 2007 or 8, in PNAS, where he identified a gene called couch potato that underlies diapause, or contributes to variation in diapause, which is a hugely important life history trait in Drosophila, and it varies in a clinal way along the eastern coast of North America. So in case you think Drosophila biologists don't have a sense of humor, well, just look at the name of the genes. The mutant phenotype is self-evident, right? So more recently, what Paul has done is create some enormous population cage experiments, which I suspect we'll hear about today, with 1 million flies in these outdoor cages that are exposed to, I'm exaggerating a little bit. He'll correct me if I'm wrong. A lot of flies that can be studied in a semi-natural situation, and this has yielded a tremendous insight. So what I like about his work is this beautiful combination of ecology and evolution with genetics. So thank you very much for coming, Paul, and welcome to the MVZ.
Thank you very much for having me. Yeah, maybe I should just stop there now. So these are the flies that I work on. No. I figured that I would go a little bit far back. I was thinking about what I wanted to talk about today, and then just in the context of having conversations this morning, I changed what I wanted to talk about 4 different times, but I resisted the urge. But I would like to talk about 3 basic things. Okay, so I'd like to talk about some fundamental aspects of adaptation, and I want to talk about temporal dynamics and the maintenance of variation, and I want to talk about genotype to phenotype translations and transitions. So just as a backdrop, so these are a group of marine snails that we used to work on in my lab when we were split between flies and marine invertebrates. I did my PhD work working in barnacles, mussels, and snails. And one of the things is just like we see with a lot of organisms is that there is a ton of variation. Here it's phenotypic variation, but has a genetic basis. And I have always been fascinated with trying to understand why populations have so much variation. And thinking about this, it was in sort of weaned on population genetics and theoretical models for understanding how variation is maintained, and getting into beyond simple heterosis to some of Gillespie's models of enzyme kinetics, to thinking about spatial variation, and then getting into temporal variation. And sort of the dissatisfaction with the level of understanding that we have, and then being okay with the sort of assertion that there seems to be some translation between environmental variation and genetic variation, right? So John Maynard Smith, there's a famous quote that's in his 2nd edition of his evolutionary genetics book that says, I'm paraphrasing now, it just seems to make sense, even though if we don't understand theoretically why, or we don't have models that explain it, it just seems to make sense that organisms that occupy variable environments are more variable genetically. Okay, so we have something like this with all this variation we see in these snails. And if we go and we try to make sense of this, so these for some of their habitats where they are, and we say, like, is there any correspondence between what snails are and what the habitat is? And we can see things like, for example, these 3 common morphs in this Littorina obtusata that match the color variation in certain aspects of the background. And we can try to understand sort of what are the processes that generate and maintain this variation in these populations if this translates down to allelic variation. Okay, so one of the things I'll talk about today is some of our sort of, well, not super recent, but recent work on temporal variation in selection pressures and how landscapes are shifting, fitness landscapes over short time scales, and what that means for our understanding of evolutionary process. Okay, and so given that it's Darwin's birthday, I figured that we could start out with something like this, and this is a, if we think about the nature of the adaptive process, one of the other things I'd like to address is what about the time scale and over which these processes occur in natural populations. So we start with the understanding that, with Darwin's metaphor of the sandstone cliff as this metaphor for adaptation, the idea that without an understanding of whether populations are mutation limited or what standing variation is, but that adaptations are something that occur by this sort of incremental or infinitesimal addition or modifications over long periods of time to produce what we consider to be an adaptation that's modified by natural selection for a particular function. Okay, and so the inherent in this sort of understanding of what this is that this takes a long time. Okay, now this also, of course, if we go through the integration of Darwinian natural selection with Mendelian genetics, we see the same thing, of course, if we go back to the classic literature. And it's a little bit like, of course, citing the Bible to support what you want, but we can find anything we want in Darwin and Haldane and everyone else. But that it seems that the calculations that we have suggest that this is consistent with Darwinian selection, that this is not something that necessarily we would be able to go out into populations and actually study. Okay? So now, of course, we do have we now know that there are lots of case studies and examples of evolution and adaptation not being over very long timescales, over effectively geological timescales. We have lots of examples of things that occur fairly rapidly. Okay? Now, one of the interesting things, this is something that I'll only touch on indirectly towards the end of the talk, is that if we do have evolution as being really, truly rapid, if it does operate on ecological timescales, then you have the opportunity for what are, sort of jargony terms for these Ecoevo feedback loops. So you can have, for example, so the example that I use for this is evolutionary rescue. So, for example, let's say that we have something like climate change, if it's real, where, let's say, populations then are depressed in terms of population size or growth rates, and then as they're sort of on their way down, they can evolve, let's say, increased thermal tolerance or range or thermal niche expansion or whatever else, which causes a population rebound. Okay, so that would be an example of evolutionary rescue, where evolution is acting fast enough to actually impact fundamental ecological dynamics. Now, also the interesting thing is that we can have this, if the ecology is directly affected in terms of population dynamics like growth rates, maximum carrying capacity, persistence, this can also in turn shape the selection pressures in these populations, which creates these potential feedback loops. So we're working on some stuff that relate to this, and it turns out if you add sort of the understanding that organisms are not a single genome, but that they're comprised of a whole bunch of microbes and a whole bunch of other things, then this becomes sort of like three-dimensional Star Trek chess, and we can start thinking about Ecoevo loops at multiple dimensions. Okay, so but this shift, this fundamental shift in thinking about evolution from really gradual, taking a long period of time where you have to essentially sit down and wait for populations to get adaptive or beneficial alleles that then have something happens to them, and then we have another, and this iterative process is now essentially that this is not the only way in which this can take place. Okay. Now, not to pick on any particular systems, Michael, but I had to put this in. This is one of the things that we always put in my lectures in evolutionary bio for some of the mouse examples. But also, I think potentially something that's worth considering is what we know about adaptation in terms of the nature of adaptation related to strong selection versus what's happening in populations that don't look like they're doing anything interesting. Okay, so arguably, one of the things that we know about timescale and about general adaptive dynamics in natural populations is strongly affected by these wonderful case studies that we have, in which we have there's the pronounced or identified agent of selection. So if we think about sort of our classic example of the peppered moth, which when I started lecturing about this, then it had all this sort of drama and controversy, and then now has sort of resurfaced as this classic example of predation, and some of Michael Majerus's work showing that predation is actually seems to be responsible for driving patterns of color variation, mapping down to the genetic level, and sort of building things back up. We can think about what's going on with Darwin's finches and Geospiza in response to drought and all the wonderful work that the Grants have done. We can think about all the stuff that's done with various mouse populations and species, sticklebacks, fungulus, and the list goes on, and snowshoe hare, jackrabbits, introgression, all sorts of things. And what arguably, for somebody who studies like something really boring like Drosophila, like I do, this is, you sort of think about this as something where you have these case studies where you have an identified agent of selection, you have links between phenotype and performance and fitness that are very concrete and well-defined, and this all makes sense. And then you look at this, and some of these, not necessarily in this case, but a lot of these seem to have maybe not a simple genetic basis, but at least maybe a tractable genetic basis. Okay, so the question is this really how adaptation works in most populations, or is this sort of, are we looking at the tip of the iceberg, or a set of case studies that are affecting how we view this in general? Okay? So for example, so if we take, this is a census size in a series of experimental populations that we have going through time. So as you take a population, let's say that's expanding, and it goes up and it hits some kind of carrying capacity, these gray lines are just replicate populations. You see that they're sort of all expanding in concert, there's a lot of variation, and then we go down to the end and they truncate. And the flies look like this in the beginning, and then they expand through these different phases, and they look like this at the end. And then you say, well, what happened? Are these fly populations actually evolving? They kind of look the same, but there's different ecological phases, and the question is, what's going on in populations like this? Okay. What is the equivalent here, or is there an equivalent for things like pollution resistance in fungulus, coat-colored mice, etc.? Okay. So given that I'm in an area that's dominated by vertebrates, so here's my invertebrate slide photo that has a vertebrate, macrovertebrate in the background for everybody here. This is also, we figured, is our best chance of doing something and getting a cover of some journals having a vertebrate in the background. So here we have some flies that we collected on a recent collecting trip in Zambia. And so what I will talk about is some of the work that we're doing in Drosophila specifically as an experimental system to look at this. So that is the genotype-phenotype translation, and think about how this is integrated, what is the architecture of these traits, what is the relevant timescale over which these processes seem to occur, and then look at whether we can relate this back to the maintenance of allelic variation in natural populations by, in this case, fluctuating selection pressures. Okay, so just a very brief background in terms of Drosophila natural history, or I should say Drosophila melanogaster natural history. Okay, so here we have any kind of like map of climate variation you could download off of Google or Wikipedia Commons. And we now know that Drosophila melanogaster originated in sub-Saharan Africa, in Miambo and Mopane woodlands, a deciduous seasonal dry forest in Africa. And then through their association with humans, has now achieved a cosmopolitan distribution that's been spread around the globe. So we have the migration events out of Africa through Europe and Asia, and then more recently into Australia, and into North America and South America. And then we have, of course, recurrent migration that's taking place worldwide, so you have the possibility for things like secondary contact zones, demographic history influencing all of these patterns of variation that we see in natural populations. So when I started working on Drosophila, the way that everyone would introduce their talks is saying that Drosophila is panmictic, that there's huge amounts of gene flow across all populations, that anything you see that looks like it has a pattern must be driven by selection. Okay, so we now know that's not true. We know that there is secondary contact, there is demography, and we spent a great deal of time, or we spent the first 10 years since I founded my lab, looking at things like spatial variation in selection pressures and phenotypes, and particular allele frequencies associated with traits, and then wondering about what time scale over which these patterns originated and were maintained. Okay? So then looking at examples of populations moving through time, right, it brought up the opportunity that we might be able to actually study this in experiments and actually observe populations evolve, as I'll get into in a couple case studies in a second. Okay. So this is what, so in a recent trip with Daniel Matute and Brandon Cooper to Zambia and Zimbabwe. So this is this is what Drosophila is in a sense native on. It's not really a host plant, but this is marula. This is in the family Anacardiaceae. It's in the mango family. This is what what looks like to be their ancestral sort of source or food source. We since found 2 other species that also, that melanogaster lives in these forest fruiting trees. Don't have pictures here. So what the interesting thing is that this is, again, not to get into too much natural history, but I find it kind of interesting. These have really tough leathery exteriors, and when they drop from the tree, these flies cannot get in. They don't have mouth parts, they don't have serrated ovipositors, they have nothing to get through to lay their eggs. So what they rely on are these holes that are drilled in here by tephridids and other species of insects, other groups of insects, that then they actually do have the mouth parts, they get through the tough exterior, lay their eggs in, and then melanogaster goes in and then sort of deposits their eggs into this developing fruit. And it turns out that the rotting rate of this marula in the field actually perfectly matches the life cycle of Drosophila melanogaster. So what they do is, so here are 2 presumably melanogaster larvae, could be simulans. I know it's not Zabrionis, but this is essentially once I break that open and look inside the fruit. So you have these larvae that have hatched out, that are now in this microbial soup of nutrients and things that are moving around. But this is a very bad place for them to pupate. Right? So once they get in, the marula then dries out as it rots, and then as soon as it gets to the point where it's dry enough, then that's when the flies are actually closing from the pupal cases, and then the fruit breaks open, and then the flies emerge. Okay, so this is all kind of interesting. So why am I telling you about this? Well, for the purposes of the talk today, the interesting thing is that even though this is a tropical environment, these are environments with very pronounced wet-dry seasonality. Okay, so this is seasonal, tropical dry forest, mostly deciduous. So this is what these habitats look like. It looks a little bit dry, it's tough to see. But these things, again, areas that are fairly dry. It turns out that it's so dry that when you collect, it sounds like a bad intro to a joke. How dry is it, Paul? It's so dry. When you take flies out from their natural habitat and you put them even in a cage, they die from desiccation in about 12 hours, even if you're supplying them with dishes. It's a very dry environment right after the wet season. Okay, now, since the expansion of out of Africa, that again follows the wake of human migration, right? And there's this interesting stuff about the commensal association between humans and Drosophilids. Now in the sort of the environments that we work in up and down the eastern seaboard primarily, also in Europe now, this has shifted to a summer-winter seasonality. Okay, so it's very different. You go from having the dry season being hot and dry with no food available to having the winter being cold and dry and not having any food available. Okay, so what got me interested in working with Drosophila in the first place was thinking about how this organism deals with this kind of temporal heterogeneity on seasonal timescales, and what does this mean? Okay. So there was a hypothesis that, actually here, let me skip through this. One of the first pieces of data that we collected that suggested that fly populations were actually moving through time on different parts of an adaptive landscape that were this temporal heterogeneity was when I sampled flies from natural orchard populations in Pennsylvania over seasonal time in multiple years, and looked at the frequency or the incidence of lines that express this reproductive quiescence or diapause, even though it's not really a true insect diapause. And so a line, when exposed to the cues, will either do it or it won't do it. Okay? So following the winter, I'm sorry you can't see this, this goes May to July to September in 2003, then May to July, September of 2004. So following the winter, this diapause incidence, which is related to overwintering survivorship in lots of different insects, is very high. It declines throughout the summer, okay? And then following winter, it's high again, and then it declines. These are samples that we collected, that I collected in the Italian market, an open-air market in South Philadelphia, that shows no pronounced seasonality, and that's what the frequency of diapause incidence was there. So this suggested to us the possibility that you have fly populations that are essentially going in this cyclical way of these life history traits that are changing over a temporal timescale. So not only do we have this latitudinal variation in terms of temperate versus, let's say, subtropical environments, but you also have the potential for these same parameters to be varying over seasonal time, and it's pronounced like summer-winter seasonality. Okay? And then so here we can see the same thing in terms of oscillations in allele frequencies for things like, for example, snips like couch potato that underlie, or at least are somewhat associated with this trait in North American populations. Okay, so the hypothesis was then that Drosophila populations, as a function of going through this seasonal environment where the fitness landscape was so discrete and so predictable over seasonal time, that we would have different peaks on the fitness landscape and that fly populations were potentially chasing this fitness peak over seasonal time. So what we would have is essentially a fitness seesaw or teeter-totter, where we go back and forth between what's good in the summer and what's good in the winter. Right? So we knew from other work that the traits that were favored during winter-based selection seemed to be associated with somatic maintenance, things like longevity, large body size, turns out darker pigmentation, things like this. And then through the summer, we would see increase in reproductive rates, faster development, and things like this. So it seemed to be this classic life history trade-off between somatic maintenance and then reproductive output. Okay? So this was the hypothesis that this was going on, and the question was, how pervasive is this? Right? Is this something that's unique to to this dormancy and overwintering, or is it more pervasive? Right? What do fly populations look like when we move through time? Okay. Now, I'm somebody that when we read things like, let's say, Lewontin's The Genetic Basis of Evolutionary Change, or we think about the quote from Haldane, or we have these descriptions, or we go through, let's say, Gillespie's book on population genetics, the understanding is that it doesn't make sense to watch populations move and evolve through time, because population genetics gives us everything we need to know about what has taken place that's important. Okay, that this sort of time-integrated patterns of variation. Okay, now, but however, if we know that there are at least some examples of populations actually evolving in real time or moving through, and you can actually observe this over short time scales, it sort of opens a door for looking at how these populations are actually changing over experimental time frames over several generations. So one of the things we wanted to do is identify the appropriate time frame over which this was taking place, and was it how many traits were involved, and what was this sort of genetic architecture? Could we map any of these things? Could we understand this genotype to phenotype translation? Okay. So we initially did this in 2 species, in melanogaster and simulans, and we were looking at sort of saying that these parameters look, when the seasonality looks very similar to things like latitudinal gradients or altitudinal gradients. So we had a set of predictions about what should change. Okay? We wanted to see whether in sequence, patterns of sequence variation, just using pool-seq at the time, whether we see the signature of these populations moving, tracking the fitness landscape changes, and then look and see what traits were involved. Okay? Okay, so essentially what we can do is we would just go through and take like these orchard populations that we work in, and then we would just go through, we would sample populations over time, and then we would bring them back into the laboratory, go through multiple generations of common garden conditions. So we were looking at genetically based change in phenotype. Okay? So this is a lot of work that was done by former graduate student Emily Behrman, former postdoc Subhash Rajpurohit, and one of my current postdocs, Seth Rudman here. And there are it turns out that there are a lot of traits that change with seasonality. So we were expecting this to be a fairly small subset of things that were based on these life history trade-offs. And it turns out that everything from body size, so some of these are correlated, but some of them are uncorrelated. So we have things like body size, dormancy, pigmentation, stress tolerance, fecundity, development, but also things like immunity, associative learning ability. So it turns out flies that are derived from spring populations are actually smarter, and they learn better and retain information longer than flies from the fall. We can call probably these like the Trump flies. Things like thermal behavior, cuticular hydrocarbons, etc. And the list goes on and on. So there are lots of fitness that are moving through time where they're evolving with large magnitude in a predictable way over at least 5 years. Okay? So we come up with this. So it's not just a single trait that's doing this. There are a ton of traits that are doing it. And all of these seem to be polygenic. Right? None of these traits, if you were to do any kind of genetic mapping on these traits, would come out as being Mendelian or discrete. Right? So the question is then for us is how the heck do you put together an architecture for a population where essentially it's moving through time over 5 to 10 generations, where all of these traits are changing, and it just keeps going in cycles again and again and again, calendar year after calendar year after calendar year. Okay? So that's what we wanted to understand. So just to give you one example for this, we can take sort of like body size, for example. So the first year we did this, so all of these are paired samples of like early, late across multiple years. So the first year we looked at this in the field, and flies, we've done this in the field, and then we've also, again, done common garden, 3 generations, eliminate epigenetics, for the most part, maternal effects, etc. There's no variation, but then all the other years, flies in the spring were large, and then they evolved to be small. Large, small. This year, for whatever reason, flies were huge. Large, small, large, small, large, small. Okay? And the same thing happened for things like learning, reproductive output, and this is immunity. The point is we have these patterns where it seems to go back and forth, where we go between summer and winter, and these traits are changing and evolving over very, very short timescales. At the genomic level, we see the same sort of thing. So this is a sort of a pre-figure out of a paper that was where Alan Berglund was the first author, is Genetics in 2014. I don't do social media. I don't have Facebook. I don't do Twitter. Actually, I have a Twitter account, but I keep getting notifications, but I've never looked at it. So this has stimulated a lot of discussion, which is interesting, and sort of like I'll get at some direct experimental ways that we can sort of try to disentangle various hypotheses for what's generating this. But so what we did is we had a bunch of samples we had collected again in the beginning and the end. Okay, we had a bunch of other ones intermediate, but they weren't sequenced. And we did spring, fall, and this was just over 3 years. And then fortuitously, we actually went out pre and post the first hard frost of the year to have one sample that was in 2011. And we have this cycling in allele frequencies over between the two seasons of spring and fall that are multiple years. And then the interesting thing about this is that in the post-frost, that we see this reversion to sort of more winter-like conditions in terms of allele frequency spectra. Okay, so this was something where, this is in a sense the sort of the first pass at looking at whether there's a signature of this. If you just do this based on binomial sampling variance and you think about how many SNPs in the Drosophila genome are going to show these kinds of patterns, it's a lot. Okay? So this was essentially taking the things that have the biggest magnitude of allele frequency change, doing something to vet them with functionality, and then coming up with this as a set to then compare with another set to see if they're actually functionally distinct. Okay, and the interpretation was these were a set of potentially seasonal SNPs fluctuating in response that seemed to be doing something different from other matched pairs of SNP or sets of SNPs in the genome. Okay? Now, okay, so I'll leave it at that, and then we'll come back to this a little bit later. Okay, now just as a quick aside, the last thing, one of the things that we wanted to before we get into what I really want to talk about, which are these direct experiments, is that we wanted to see whether any of these seasonal SNPs actually seem to be functional and seem to do anything interesting. Okay? So this was, in other words, we come up with a very simple hypothesis that alleles that show these kinds of patterns as a set, are they doing something that's qualitatively different in terms of functionality to things that don't do these kinds of cycles? We wanted to test a handful of these to see whether they had any effects on traits that we knew varied with seasonality or with time. Okay? So this is basically me hand curating a couple of examples. So I'll just go through just one really quick example. I think in the long run was kind of misleading in terms of what we wanted to get out of this. So one of these, so I'm basically looking at these lists of seasonal SNPs in the genome that Alan had generated with this pool-seq approach. That was this collaborative with Dimitri when he was a postdoc in Dmitry Petrov's lab. And we have this list of seasonal SNPs. We have stuff that varies with latitude, and I went through and basically hand-curated it and came up with a list of things I thought looked the most interesting, where we see coincident patterns of seasonality and clinality. Okay, that is where the frequency, where the SNP that's high frequency in the spring is the one that's high frequency in the north. Okay, not things that show discordant clines. Interestingly, there were 5 things that came out that were all trip channels, which at the time were being used. This was before they were sort of used as Drosophila in the Drosophila toolbox as temperature-sensitive things, but they were identified by Craig Montel's group initially in Drosophila as being genes that regulate temperature sensing. Okay? So these are the kinds of things where if you knock out a particular trip channel, the larva doesn't sense that it's being probed by a cold needle or that it's on a hot plate. Okay? So it regulates temperature sensitivity, right? So seasonality has something to do with temperature, so we were like, this seems to make sense. Like, why don't we take a look at a couple of these trip channels? Okay, so what we decided to do was basically take summer and winter alleles, and in the sort of the pre-CRISPR sort of days, what we would do here is sort of the poor person's transgenics or to phenotype SNPs, is we took SNP states, essentially, and took a bunch of lines that had the same SNP state, let them all recombine, did this in replicate with independent sets of lines, and made up other cages that had the alternative SNP state, multiple replicates with independent lines, let them recombine for 10 generations, the idea being that the only SNP in the genome that's fixed is the one of interest, and everything that's linked to it, but it won't have an FST of 1, it won't actually be fixed. So we can show that bioinformatically that effectively that this is a way that you can sort of compare this cage to this cage, and the only difference between these outbred populations is this one SNP of interest. Okay? And then we do this in multiple experimental replicates and sort of take a look at these. Okay. So it's a first pass, but it's not super satisfying. So we built this temperature chamber to look at thermal behavior in smaller insects, and then we can also do things like field releases, and you can do UV dust on these things. You can put them in different thermally heterogeneous things in the lab versus in the field. This is the first version of our sort of experimental orchard. This is outside of my old lab. This is now no longer there. There's another huge building there, so we had to move. And then you can do things with like infrared imaging, and you can come up with sort of a thermal landscape, and you can look and see where flies like to go, and look at whether the trip channels seem to be influencing what they're doing in terms of thermal behavior. So I had a rotation student, Justin Walsh, who did all this work in the field and just did a fantastic job. He's a behavioral ecologist. And so what we ended up doing was taking, so at this particular trip candidate, this trip A1, we basically have a winter allele and we have a summer allele. It's just defined in when they're at high frequency. And we're only looking at homozygotes because these are the flies that are coming from those recombinant cages. Okay? So we have the winter in blue and we have the summer in red. And we looked at this in the thermal gradients, okay? And you bin this in terms of like high temperature, intermediate temperature, and lower temperature. Right, to our surprise, what we essentially see if we just focus on this over here is that the flies that are carrying the winter allele, this is in replicate independent populations that have independent genetic backgrounds, that they avoid being in the hot area, and the flies that are carrying the summer trip allele tend to prefer that. We were like, okay, well let's check that out a little bit more. So we did thermally heterogeneous cages in the basement of our laboratory, one of our basement labs, so we can do all sorts of crazy stuff and no one yells at us. And we put in things like buckets of ice and heaters and fake trees and real trees and made a thermally heterogeneous environment, took the imaging to see where the flies are. We see the same basic pattern, right? Summer alleles prefer, the flies that are carrying these alleles prefer warmer temperatures. Flies that carry the winter allele prefer the cooler temperatures. In a bunch of field releases outside in our field mesocosms, where we're not controlling anything, but we just use the natural ambient temperature and the variation, we see no difference at the lower temperatures, but we still saw this difference where the sort of sets of cages were behaving differently in response to high temperature. Okay. So this suggested to us, naively, that we could potentially make sense of this sort of genomic signal and try to understand causation versus things that are getting dragged along. Okay, so that's where I want to go. All right, so this is, so the first thing we have is this potential here for trying to make sense of this kind of a dynamic is to make it very simple and just make it binary. So we have essentially population growth through the summer, and then we have response to winter, okay? And then the number of generations is sort of up, is for debate. It looks like we have something else going on in its pre-winter phase, but we have differences in terms of number of generations, and we have this basic switch between reproduction and then somatic maintenance. Okay, so the question is, could we do this experimentally and try to get out of some of the limitations of doing sampling of natural populations? And I'm an experimentalist at heart. Let me see if we can do this using experiments. Okay. So this is what we have as looking at these fitness trade-offs. The first thing that we wanted to do, and this is a while ago now, is to look and see whether we can simply have fly populations go through expansion phase. We thought like a summer, and just make it as relaxed selection as possible and have these populations expand. And do we see a signature of this kind of expansion? And then go through something that's really hard in terms of somatic maintenance and stress tolerance, and basically go through a survivorship curve and see if we see this rebound. So the prediction is that we would have some sets of things that would increase in frequency during population expansion when selection should be on reproduction and development rate. And then we would see the reverse during when we expose them to things like temperature stress or starvation stress or desiccation stress or something like this. Okay. All right, so this here, these are slides that actually are borrowed from Sharon for her nice depiction of the experimental design. So Subhash, my former postdoc, and Nicholas, who was an undergraduate who was Subhash's shadow for like 4 years in our lab, did this crazy experiment. And then, as I'll get to, Sharon did all the really hard computational and wonderful analysis on this. So what we started with was a series of inbred lines, expanded them out. Basically, all we could fit in our basement lab was 4 replicate cages. So we did this, and we basically went through, we doubled the amount of food that was produced every generation, and we went through a series of generations until we basically couldn't fit any more food into these cages, and we got the flies up to about $600 to $800,000. That's $800,000, yes. 800,000 flies per cage. That's probably how much it costs. And then we go through, and then we didn't even do this about phenotypes. We just looked at the profiles of allele frequency change during this expansion. So that's one phase. And then we would look and see what's taking place over this trajectory, and then we would essentially do the selection on the opposite, and then we would repeat this and do this 2 different times independently. Okay. All right, so this is you have to forgive me. I took this with my old iPhone, like my iPhone 4 or whatever it is. So it's hopefully a thing, but that's what I can't remember what this cage was. We did this with volumetric measurements. So after all the flies died during the starvation desiccation phase of this, we would just vacuum them up and we would sort of do volumetric things and estimate how big the populations are. It's a lot of flies. So I think that this is the way. Yeah, and this is where you could actually hear them sort of going through space and stuff that it was getting. And everyone in the department was complaining about all the flies everywhere. And I have no idea what you're talking about. So this is the analysis that, so Sharon, I shouldn't even put a picture, I could just point to you and say, like, Sharon, this is what. So we have this, our 4 cages. So if we look at this in terms of these multidimensional, these MDS plots with pairwise FST, you can see that we had some issues in terms of our population founding. So we have one cage was really different from some of the other replica cages, even though they were supposed to be the same. Okay, but so you can see that we do see a signature of cage by cage in terms of our 4 replica cages. The question was whether we have enough replication to actually say something meaningful. So here we're going through this. These are 9 generations of expansion. Okay? And so now what we're saying is taking these same points in space, and what Sharon has done here is plotted these, now they're color-coded, by generation. So these are time points as we move through generational time. And what you can see is that on this axis, right, which doesn't have axes, it doesn't have values, it's unitless, that we're essentially moving in a way that's consistent with time. These populations are moving through this in a way that is temporally consistent, going from one time point to another, right? Suggesting that these populations are evolving in a way that's predictable, right, in terms of this pairwise FST over this generational time, right? Then Sharon would do the permuted data to see whether this pattern is something that we would get, right? And this is the permuted data, right? You can see that these patterns look very different. Okay, now the real cool thing that came out of this was to look at what happens during the second phase. Okay, so what we did, this is the really crazy thing where we used to tag team. So we would do the fastest selection experiment that we could do would be to have them die over 24 hours where we remove food and remove water. So that means that the 3 of us were up and we made these vacuums with these long aspirator things. So we would sit there and then at different time points we would estimate how many flies we think had died. We would go into the 4 replica cages and vacuum up the living flies as they were sort of dying through the survivorship curves. And we did this, it took us about 32 hours straight to do it, which was did not endear me to anyone in the lab. But I did participate in that, I would like to point out. So here's what we see during the expansion, again, to remind us of what these data are across these 9 time points, which are 9 generations moving through time, right? And this is what happens during the starvation phase. Right, so these are samples at time 0, 6, 9, 12, 15, 19, 21, 23 hours. Okay? So these are just like the generations. Now we're talking about this as in hours, not generations. And what this is as these populations were moving through space, and essentially on this MDS plot, through generational time, they essentially reverted along the same axis, or along the same diagonal, in a way that is actually now also consistent with time, but in the opposite direction. Suggesting that is this potentially this underlying set of things, probably blocks of linkage disequilibrium here, but certainly something driving this, that's essentially moving things during the MX phase, and it's the same set of things that are moving this during the LX phase, right, during selection on reproductive output or during survivorship. Okay, so one other thing, you said stop at 1, so I have like 10 more minutes? All right, I'll get through this, no problem. All right, so now the last thing I want to say is about one experiment that we did that now, this is something that's telling us about potential fitness trade-offs and whether we see the signal in a laboratory environment. What does it look like when you do this in the field? Okay? So we built this orchard. You have these field mesocosms. We have like 60 of these. I'll show you some pictures. It's basically just like an insect cage, and we have a peach tree. We have shelving units and webcams just for fun. Things like, to keep track of temperature, humidity, whatever else. Okay, we've done a whole bunch of experiments since 2014 when we built this and we got this extra space down. So Penn bought this old, wait, I'm getting recorded, right? So, used to be an old chemical plant that was then deeded over to the university, and the chemical company gave the riverfront property to the city of Philadelphia. Penn bought the rest, and then the company's absolved of any remediation of the property. It's an industrial brown site. It sounds like a great place to do field work. So we went down there and built this experimental orchard. We've done a bunch of different things. Okay, so we've done things like, let's see if we can mimic climate change and increase temperature. We can basically try to break up this Ecoevo dynamic. We can look at things like parasitoid load. We can look at competition. We can look at trophic cascades. We can do all sorts of things, manipulate migration, the source of genetic variation for Africa versus Florida versus Maine versus Pennsylvania. We can do things with chromosomal inversions. We can do all sorts of things. We've done microbiome manipulations. But what we've done every single year is just basically take a series of Pennsylvania populations and just culture them and move them through seasonal time and take samples and sequence them. Okay, so this is what this started out as when we got all this first shipment of peach trees and we built the fence. And it looks like this now. It still looks a little bit industrial. This is the last of our phases. The rest of our 60 cages were getting put up. And we have our climate tower, and then all these are these, this pressure-treated base where we have the stainless supports, and then we have these zippers. And we've also, this is a question that comes up a lot, is whether we've tested whether we have cage integrity over time. So we actually did both. We did a positive control of this where I accidentally released flies from one to the other. And then I didn't tell Sharon about it, and then she came back and she said, "What happened with that one cage?" And I said, "Yes." Exactly. Okay, so then what we can do is, so here's Seth and Ozan, Ozan in my lab, and Seth is a postdoc. So each one of these cages, what we can do is take flies, do whatever we want to them in the lab, we bring them back out to the field. So these are, each one of these cages represents, this is 2,500 flies that are equal sex ratios, that are all density controlled from the same exact cohort. Which means that we have to keep a lot of flies in the lab, and we collect generally about in the order of 50,000 flies to then bring to the field in these little cages, and each one of those little cages goes into a big cage, and then we expand it out through generational time, right? So we bring this down, hope that we don't, we have to be careful not to roll them over and have flies die. Put them out in front, release them, and then we just feed them over seasonal time. Right? The populations do get, here's poor Nicholas, who started wearing full-body mosquito suits because the fly populations do get extremely large. And all we do is we feed them lab food. Okay, so we want to make this simple. So it's all peach trees, but it's also, remember, it's on an industrial polluted site, so we can't actually eat anything there. So we pick all the peaches, throw them out, and then we feed them Drosophila food. They lay eggs on the substrate, and then we basically put cotton rope around it, put a lid on it with mesh. We count the number of eggs that are laid, and we do density, we do adult density and census size every week. So we keep track of sort of the demographic stuff of the populations, and then we keep track of their larval input and everything else. So this is, again, apologies, this is me taking stuff with my iPhone as I'm walking around. I think I turned the volume off good because I probably have commentary about what happened here, we did what. But you can see that in these cages, right, so this is just where we culture the flies, but it's tough to see with this, but there are lots of flies that are flying around. All right. Okay. How can I get out of here? Yes. All right. So the design here, so for this experiment, that this is basically, we're not doing anything to these populations, right? This is the boring experiment. We have seasonal populations, right? All we're doing is we're seeding these cages with they're density controlled, they're from the same exact cohort, they're mixed sexes, from the same local Pennsylvania source population, and then we're basically letting them go through time. Okay? So we let them go through time, we feed them, we keep track of reproductive output, we keep track of census size, and then we take samples, right, on a regular basis, take flies, bring them back to the lab, go through 3 generations, do common garden phenotyping, and then do sequencing. Okay? So these experiments, depending on which experiment, takes place over 90 to 160 days, if you're really being masochistic. Okay. So what are these? So the first thing is what happens to phenotype. Okay. So this is from 2 years where we did essentially something similar but over different timescales. Okay. So I'll just be talking about the 2014 genomic data here. So this is heat knockdown. This is just a response of heat. It always tends to be messy. We have starvation resistance. We have reproductive output, and then we have desiccation tolerance. Okay? And these are different time points as we're moving through time, so what our founders were in blue, and then our time points that were sampled in July, August, September, and October. Okay, so it's a fairly truncated time frame here. In this particular experiment, this is going from, the founder was on July 15th, and we're only going through October. Okay? So this is on the order of, let's say, something like 7 Drosophila generations, 7 to 8 over this time. So the amount of phenotypic evolution, right? So, and then the means are plotted in the red, and then the gray lines are the replicate cages that are from the same population that were found by those different little cages. So you can see that phenotypically there is at least some degree of parallelism. Sure, there's some spread, but the populations, by and large, seem to be doing the same thing phenotypically. Okay? The same thing happened in another year experiment where we did something with slightly different source populations, but you see these populations evolving as we're moving through seasonal time. Okay? These, by and large, match the patterns that we observe in the natural populations, suggesting, for example, that starvation tolerance as you go through the summer declines. Okay? If you do the experiment long enough, the starvation declines, and then it goes way back up toward in the winter at the end of November. Okay? But here we only did it to October. And then fecundity during the summer, for example, increases rapidly as well. That goes from, this is going up threefold over 7 generations. Again, so this is common garden. This is all genetic change in the populations. We've eliminated almost all the epigenetic possibilities here, going through 3 generations. I don't want to exclude that as an entire possibility, but it seems like we've eliminated most of it. Okay. Now, just to give you an example of what some of these things look like, so here's an example of the evolution of pigmentation, which is another story that I'm not going to talk about. But so, initially flies in the spring come out from winter, and genetically they're dark and they're large. Okay. As they move through seasonal time, they evolve to be lighter. And it's tough to see in this, but you can actually show that there's changes in tone as they get more sort of yellow, and we sort of know a lot about the cis-regulation of these pathways and everything else. Okay? And then as you get into the fall phase, some of these things, depending on what we've done with the density and other things, then we'll re-evolve this dark pigmentation by the end of November. Okay, so again, these are populations that are sampled, taken into the lab, go through 2 to 3 generations, depending on who's doing it, what experiment, right, and then are phenotyped. Okay? So this is not plasticity. This is all genetic changes, okay, in these populations. This is incredibly rapid. It happens over and over and over again. And this is just one example of one of the 9 traits that we've measured. Okay, so what about in terms of patterns of genomic change? So this actually, Sharon, you're the only one who knows this. I pulled the wrong data set. This is from the other year. Okay, but so these MDS plots again that Sharon constructed in her analysis of this. Again, this work being that this is a collaboration between my lab and Dmitry Petrov's lab at Stanford. And this here again, we have these ellipses around these cages that are moved. The ellipses are around different time points. Okay, so in this example here, which is an oversimplification based on what this actual data is, we see again this temporal signature of populations moving through these multidimensional scaling plot space, right, where we actually see that they're evolving over time. Right, there's a consistent track of time and of cage. Okay, so this is telling us, and I'm going to skip through this in the interest of time, and go to what we don't like, which makes everybody nervous. Especially Sharon, are these Manhattan plots to illustrate the second point. So if we look at whether these things are actually evolving. Okay. So this is now based on an effect size and an empirical FDR in terms of the color pattern with for this over different molar elements. And this is from time point 1 to 2, time point 2 to 3, 3 to 4, and then 4 to 5. Okay. And so at each time point, right, there are stacks, or there are outliers that both have the effect size and have this empirical FDR, right, that suggests to us, and then in addition to, in conjunction with other data, that these are not simply driven by drift, and this is not noise, that we do see that there are patterns of enrichment, this window-based enrichment. Now, the interesting thing for me is thinking about this in terms of the landscape, but also what about time point to time point? So interestingly, as you go, so at time point 1 to 2, you can see that there are some things that are changing over time here that match these criteria. But when you go from time point 2 to 3, right, none of these that were initially found in that panel are actually coming up here. When you go from time point 3 to time point 4, again, the identity of these is not consistent from time point to time point. And the same thing happens when we go from time point 4 to time point 5. Okay, so as we sample this. So at each time point, you're seeing a signal of something happening genome-wide. That's happening on all chromosomes. It's not driven by chromosomal inversions. It's not driven by things, the dynamics around centromeres or recombinational landscape exclusively. But the identity of what's changing is so dynamic that it's actually changing from time point to time point to time point. So we've identified by 5 samples over, in this case here, over a 100-day experiment, we essentially have not identified the appropriate timescale for the resolution over which these are changing, right? Because by the time we've taken the sample, it's already shifted. Okay? So this is the entire time frame here. Again, we're talking about something in the order of 10 generations. Maybe a bit more. Okay. All right, last thing to say. What does this have in terms of getting back to the sort of integration of ecology and evolution? What does this say about, what does evolution, this rapid evolution, tell us about basic ecological parameters, things like demography? So there's a trick that we can do in these experiments where we can prevent evolution from taking place. Okay, what we do is we take a set of cages. These flies, they lay eggs. We count the eggs. We throw them out. We take the same number of eggs from the ancestral population that's kept in the lab under conditions where we don't let them evolve as much as possible. And we keep this going over and over again every 2 days. So what we're doing is preventing population from evolving to the field conditions as much as possible by replacing us with the ancestral population. It's the best that we can do from going to the freezer and getting frozen stocks from things, and we can't do that. Okay? So the interesting thing is, again, these 2 replicate year experiments where we do the same thing and just culture through time. If you let fly populations evolve to the seasonality, they basically start off at a little bit of a disadvantage, at least in this year, but they achieve a higher carrying capacity, right? And then they persist longer through time than the cages these ones that are not evolving, that are still genetically diverse. Okay? The same thing happens when we run this experiment with a different set of populations in a replicate year. So that what we're seeing is this first part of rapid adaptation providing demographic benefits in terms of what's happening in terms of population growth, persistence, and then in maximum size. Okay? Okay. Now, what about, so the other thing we did is we manipulated the amount of genetic diversity. Okay? So in this plot here, what we have is days from the start of these experiments. The orange line here are the populations that are not evolving. They're still genetically diverse, a ton of diversity, but they can't, and we prevent them from evolving to the field. They're still probably evolving to the basement conditions because they're an outbred population, right? But we're preventing them from evolving to the seasonal environment outside. Okay? And then what we did is we took an inbred line, we took 1 isofemale line, 5, 10, 25, and 50 isofemale lines, and looked at their demographic trajectories. And what we see, so here is census size at day 23, here it is at 53, and here it is at 120. At each time point, basically all it takes is 1 isofemale line to have a demographic benefit above a genetically diverse population that's not evolving to seasonality. Okay? So this is suggesting to us, and you can see this in terms of what's going on at all 3 time points during the expansion, during this density-dependent phase, and at the end of the experiment. Okay? So rapid evolution, not only do we see this in terms of multiple phenotypes changing through time, right? These are lots of fitness traits that are doing this sort of a seesaw thing and rapidly evolving. We see genomic signatures of the same thing taking place. It's incredibly dynamic from time point to time point. And also this has fundamental, this has this output in terms of fundamental aspects of population growth and aspects of population demography. Okay? So this rapid evolution is actually doing something in terms of ecological dynamics in populations. Okay? So just to summarize then, what we have is that if we think about this, right, this is not just rapid, this is really, really rapid. Like to the point where I don't know if we've identified how quickly this takes place, but we know that it can occur as fast as 2 to 3 generations. Okay? So we have the possibility that basically we see stuff going from parent to offspring where actually the parents are being selected and their offspring are experiencing a completely different environment, or at least sufficiently different to drive these patterns. Lots of traits. These evolve predictably and also in parallel. Okay? Suggesting that it's not just rapid evolution, but it's rapid adaptation, okay? Which is another ball of wax. But this does appear driven, so the indoor cage experiment and then the stuff that we have outside suggests that this sort of seesaw fitness dynamic may be as simple as being generated by these fundamental fitness trade-offs between reproductive output and then somatic maintenance. And that might be simple enough to create this kind of dynamic over these time scales where again, the fitness surface is fluctuating. Okay? This is strong enough, right? Even though we don't see this sort of smoking gun of the particular polymorphisms or things like a transposable element, something popping out as being driving these patterns, it's strong enough to cause this, and it's genome-wide, and it's over very short time scales. So this is essentially polygenic. Might not be omnigenic, but it certainly seems to be polygenic, right? And we see these parallel frequency shifts occur here on all chromosomes and potentially on all Muller elements. And then getting back to where I started in the beginning of the talk, right? So this really does suggest that getting this together, at least in these experiments where we can know that it's not driven by migration, we know there's no cryptic population structure, there's all the things that plague our sort of sampling of natural populations, we can eliminate with simple experiments and replication over seasonal time in this orchard. Okay? And this does suggest that even though the temporal models, the models for temporally variable selection, maintaining variation, might not be developed to the point where this is all consistent, it does suggest that this might be a mechanism by which some of these alleles are maintained over long periods of time and don't go to fixation. Okay. And so with that, again, I'll stop here and say again, so this work is in collaboration with Dimitri and then Sharon and Alan Berglund, Heather and Suzanne in Dimitri's lab. Also Jonathan Levine and Mark Turcotte and other people in the Levine lab at Princeton. And then people in my laboratory, and then funding sources and some consortia where we're doing all this sort of sampling and fun things in different continents. Thank you very much.
Maybe we'll let people sneak out who have to go to classes, but I'm sure Paul would be happy to take questions.
Yeah.
Can you, because evolution is taking place over such short timescales, can you sample individual generations?
We have not up until now. No, but we will. So the way that we culture these in the field is overlapping generations, so it's not discrete. So these are just conveyor belts of things being produced. So we sort of go in, let them lay eggs, trap, do this, put it in cages, release the flies, you know, sort of go through this whole rigmarole of this conveyor belt. But we do want to do more dense temporal sampling. And then the other big thing for us to do is to shift from doing everything based on a pool-seek approach to trying to look at things on an individual basis, which was cost prohibitive, to be honest, when we first started this back in 2014, but now is a little bit more reasonable.
So when you do sample, it's the possibility that you're sampling multiple generations in a single sample.
Absolutely.
Yeah.
Yes. Yeah, and so we've also done some selection, or some experiments on lag load, and looking at single generation versus integrated across generations, and have those sequenced as well, suggesting that so we've done single cohorts of like smaller cages within the bigger cages, and looking at that, how that changes. So we have tried to see the patterns for those single generation experiments relative to what happens when we just go into the cage and sample, and they're fairly consistent. Yeah.
From the couch potato example, it seems like the architecture of seasonality is very pleiotropic, but in this genome shift case, there are so many other regions. So what kind of role is the couch potato play, and is that co-opting with these other seasonal regions as well? Yeah. So the simple prediction would be that that we would have the things that are fluctuating seasonally in the natural populations are the ones that are changing in the orchard cages, and there seems to be enough sort of variation going from year to year that we're sort of having a difficult time narrowing in on a set of things that are consistent across all years and in all experiments. We do have a lot of traction in terms of using clinality as a sort of functional vetting to look at these patterns to see how things are sorting out with space. So if you think about what the patterns are for case-by-case things, one of the most interesting things for me when we started doing this is all of the canonical examples that I knew about in terms of adaptation in flies are not in the tails of any of these sequencing things. So, for example, if we look at alcohol dehydrogenase, or if we look, couch potato actually does tend to fall in the tails actually a lot. So I'm just putting my foot in my mouth. But other than some of the other ones, so it's definitely integrated within a whole bunch of things, and I don't know how these are all put together to affect these traits. We do know, right, so what we would have to do, and this is, again, yeah, this is anyway, this is the subject of a grant application that's going in, but what we need to do is go into these cages, take flies out, pre and post or at different time points in the rapid evolution, and then actually do quantitative genetics on them to understand the architecture, to really know how these things are put together. So all we have right now is sort of just this snapshot of going in and we just sample and then we just phenotype and we do pool-seek, but we haven't integrated it to sort of do really hardcore cis-regulatory experiments in the case of some of these pigmentation, for example, or to do G matrices at different time points and how all this fits together. So that's what we're trying to get to, but we're not there yet. Yeah.
Thanks for the really interesting talk. I had a question about the study with TRPA1.
Yeah.
The winter and summer alleles. I was curious, do you have any information or predictions about how you think the channel might, is it even modified between the two different alleles, or?
Yeah, so so let's see. So I think in that, so we know that there seems to be differential expression. So those SNPs are upstream, and those particular ones that we sort of,
Now, those SNPs that we're using to interrogate this are just probably acting like markers, right? Like, I'm not saying that they're causative. I'm just saying that there's an allele there that tends to it's probably like a linkage group or some kind of like block of LD. So I'm not saying that these are causative, but it definitely seems to be associated with this, and they are differentially expressed, but we don't have, so like what we would want to do if we wanted to get at sort of mechanistic stuff would be to try to go in and look at different things and whatever in particular places and see if that matches with like single cell RNA-seq or something like that. I think that the outcome, it's like the reason why I keep it in there is because it's just like a nice little sort of like makes me feel good story about like that we actually made some traction. But I really think that when we look at sort of the polygenic nature of this response, that going in and trying to make sense of what's causative and what's driving stuff is really, I just don't think we're there. I don't think we really know, like given these surveys, if we can go in. So the problem that we had with the TRIP and the other ones, we did it with the same thing with like 10 other polymorphisms. Every single polymorphism we phenotyped had huge effects on the traits of interest and not on control traits. So like if we did a circadian gene, it had effect on like tau, or like some aspects of rhythm or periodicity, but then it doesn't have an effect on heat tolerance or reproductive output. Then we look at TRIP channels and they have effect on thermal behavior, but not on heat knockdown. Or actually, no, they do have it on heat knockdown, but not on like fecundity. So then we're just like, how is it that all of these things can be so functional where it looks like all of them should have a selection coefficient of like 0.5? So then like that's where I was sort of like, okay, maybe we should take a different approach to looking at this. So that's why we didn't go down that road further and trying to understand what's going on with that, but it would be really interesting.
Paul,
Yeah.
just wondering if you could speculate a little bit on how general you think this might be. So Dobzhansky found seasonal polymorphisms for inversion frequencies, and you see it in Drosophila. But Drosophila have a lot of generations per year, and presumably for things that have a generation time of longer than a year, you wouldn't expect this sort of pattern. For things that have a generation time of a year, you might not.
Yeah.
How general do you think this is going to be?
One is so given that I'm an empiricist and an experimentalist, I feel like that's an empirical question, right? So I would like to extend this out and look at other systems. So again, like there are multiple ones in which we could do this with 2 generations a year that have like summer-winter phasing or have multiple things in different contexts. So the question I think is whether this microcosm of seasonality is just a way in which we can compress the time scale. So like let's say like from large vertebrates or something, it's just the stuff that we're seeing, is it just that the temporal axis is different? And we have such pronounced differences here that might be played out over longer generational time for things that have, again, like 1 generation every 20 years or something like that. So I think that that's an open question, but we don't even know whether these things happen in other organisms that are multivolti. So I think that that's the first place that I would like to start, of things that have multiple generations per year, or at least have something associated with seasonality is just a window into this, and see whether these general, so I'm predicting that yes, we will see that. And then the question is whether that extends out in terms of population genetic integration to other things, and what is the appropriate time scale, right? So I just think that it's basically, the basic point is that if you do an inference based on a variation in polymorphism and divergence, basically what we have is what we have now for variation, but then we have divergence, and we have 2 points, and we're drawing a line. And this is telling me that there's a lot of stuff going on in between the 2 points. And so I think that there are a lot of idiosyncrasies, but I'm obviously very biased, but I do think this is saying something general about the evolutionary process, that it's far more dynamic over different timescales than we appreciate it.
Hi. I have to express a question that's not extremely well formed, but like I will try to do my best. So if you have seasonal SNPs, let's say for the summer, and it is like 100 SNPs, and you collect like a certain population in the summer, do you expect these 100 SNPs to be found like in all the flies of the summer, or could it be that like 30 of these SNPs are like in a subpopulation, and then 30 are in another, and 30 are in another, and then actually if you can like separate them, then you may see a pattern emerging.
Yeah, it's a really good approach. So if we had so we tend to not have a ton of private alleles in Drosophila populations, so most of these things that are polymorphic are actually screened to be polymorphic in global populations, so that we're not getting all these things that might be like present in one but not the others. But I do think, as a general approach, so one of the limitations of what we've done so far is that we really have only done local Pennsylvania populations, right, and really focused on that. So, for example, if we take what we're going to do this summer is we have ancestral African populations that we just got from Zambia. So we're going to see, like, do they have the same kinds of seasonal dynamics that Pennsylvania populations do? We've done introgression from like Maine and Florida into Pennsylvania to see how that affects the dynamics. And it turns out that it just looks like if you get variation from anywhere, that it actually speeds up your rate of adaptation. It doesn't matter where the variation comes from. But we have all these things like the Jansky-Muller elements that seem to be segregating within populations and all sorts of crazy dynamics. So I don't know if we have the independence to sort of test whether we might have subsets of things that we would get a concerted pattern. What we are trying to do is with the Drosophila consortium is to do this sampling in Europe, and then we're now extending it to South America. We're trying to do it in parts of Asia, so we can have populations that probably are really different in their allele frequency spectra, and we can do this as independence and see. The point is, I think that this is a power issue, and I think that we do need to have, it might be potentially that we need thousands of samples of paired sort of seasonal samples to actually get at this distilled core of what actually is predictable. But I don't think we have enough power to do that yet, and that would be exactly a good way to do it.
Maybe we'll stop there, and if you have other questions, please feel free to come up and thank Paul one more time.